All right, we'll finish our stains in this last video for chapter 2. Our next type of differential stain is the endospore stain. And endospores are protein-rich structures that allow certain bacteria to survive very harsh environments. Not all gram-positive bacteria can make endospores. It's a certain subset. Specifically, the bacteria from genus Bacillus, from the genus Clostridium, and Clostridioides. And we'll learn about at least one other group later on. These are examples of some bacteria that can produce endospores. But the gram stain is not good at detecting endospores. Endospores resist staining, so if you just do the gram stain, the endospores will be clear. The most standard procedure we use for the endospore stain is the Schaefer-Fulton method, in which we're going to steam the primary stain, malachite green, to force it into that thick protein layer of the endospores. The decolorizer is the water step. So you're just going to use water to rinse off the cells or rinse off your smear. And malachite green will be removed from the cells, but not from the endospores. So these will stay green. Finally, our counter stain will be safranin, the same safranin that we used in the gram stain, and that is going to stain the rest of the cell pink. So cells will appear pink, and if there are endospores present, they're going to appear green. Here's another look at that endospore stain. Remember, cells are transparent before we stain them. The first step, malachite green, that primary stain, stains everything, so cells and endospores. Decolorization with water will remove malachite green from cells, but not remove it from the endospores. And then the counter stain, safranin, will stain cells, but not the endospores. The endospores will appear green, the cells will appear pink. Endospores can be present within cells. Sometimes they're going to be found outside of the cell if the cell has already released the endospore. This picture shows you what you're going to see soon once we start doing the endospore stain in lab. You'll see red cells, or kind of pinkish cells, with green endospores. The endospore stain is great, but there are some drawbacks. One is it takes some time, so you need to steam the malachite green, forcing it into the endospores. Another is that when the cells are young, they might not have very many endospores. So older cells are much more likely to produce endospores. And older for bacteria means like 2 or 3 days old. A week old, you'll have lots of endospores if these are endospore producing cells like Bacillus or Clostridium. A faster way to identify if endospores are present is often through phase contrast microscopy. And you can see some images here. If you do phase contrast, you'll be able to see that the endospores really stand out. They're kind of like a white, almost like a Tic Tac candy, if you've seen Tic Tacs before. And I can see them over here as well. So endospore stains are important in identifying certain bacteria, such as Bacillus. Bacillus anthracis, which causes anthrax, is of particular interest because its endospores can be used as a bioterrorism agent. And in fact, there were a series of bioterrorism attacks caused by these anthrax spores in 2001, right after September 11th. So what happened were letters containing anthrax endospores were mailed to different news offices and Democratic senators, and they were disguised as if they were coming from a kid. Ultimately, it led to about 22 confirmed cases of anthrax infection, as well as 5 deaths. And they ultimately traced it to an individual, Bruce Ivins, a scientist at the government's biodefense labs in Maryland, that led these attacks. So is endospore staining positive, negative, or differential staining? What do you guys think? Remember that it's differential staining, right? It uses 2 different stains, malachite green and safranin, to be able to tell apart endospores from regular cells, vegetative cells. So endospores are going to retain that malachite green. It'll be washed away with water in regular vegetative cells, and the vegetative cells will take up that counterstain, safranin, and they'll look pink. So this allows us clear identification of the presence of endospores as well as where they're located within the cell. We'll learn more about that part later. and it helps us narrow down what kind of cells we might have, right? Do we have bacillus? Do we have Clostridium? Do we have Clostridioides? If something doesn't have endospores, we'll be able to say that it's probably not in one of those three genera. Our next type of differential stain is staining for flagella. And flagella are these really thin tail-like structures that cells use to move around, right? They're for motility or movement. Bacteria have them, archaea, and eukaryotes, but not all cells, so certain cells within each of these categories. Flagella are too small, too thin to be seen with just basic microscopy, so they need this special staining if you want to see them under the microscope. And this usually involves a step with a mordant first that is applied to coat and thicken the flagella. That will be followed by staining, so you can see color. And a common stain shown here is para-rosaniline, or we can also use basic fuchsin. Staining will allow us to see how many flagella a bacterium has or a cell has, as well as their arrangement. And we're going to learn more about these names later, but bacteria can have 1, 2, a few, or even a ton of flagella that coat the entire cell. And they're important in identification of what kind of cells we have. But we have to be careful when staining because flagella are very fragile and really easy to break. So when you're actually going through the staining process, many of them will break off, but you should still have several that you can visualize under the microscope after staining. This example from our book shows these flagella very nicely surrounding this rod-shaped bacterial cell. You can see the flagella are surrounding the cell. The chart in our book gives us a nice summary that allows us to compare and contrast these different stains. Remember, we have our simple stains. These usually involve just one step or one stain where everything's the same color. And I wanted to make a note here for acidic stains. We said that these stains have a negatively charged chromophore. Our book, I'd like to let's stick to negative for now to make things more clear. Although I mentioned earlier, sometimes acidic stains can bind to some organism depending on the organism's chemistry or the pH of the environment. And on this and the next slide, we have all of our differential stains. Remember, these involve 2 or more different types of stains. They're multi-step, and they let us identify specific structures on the cell or within the cell. As you go through the differential stains, make sure you know each step, as well as what colors positive and negative cells would appear at each step. For instance, in the previous slide, the Gram stain, what's the first step? Crystal violet, so all cells will be purple. What's the second step? Iodine. They're still purple. Third step, decolorizer with alcohol. Okay, now Gram-positive cells are purple, Gram-negative cells will be clear. And then fourth step, safranin. Gram-positive cells will still be purple because purple will mask the pink since purple is darker than pink, but Gram-negative cells will appear pink because in the previous step they were clear. So do that with each of these stains. Make sure you know each step, the names of the stains, and the colors the cells will appear. So I cut out a lot of the end of the textbook in my slides. I'm not going to go over the check your understanding questions for some of these, but I did want to bring up some aspects of the last part of 2.4. One is an ultramicrotome. So before I talk about the ultramicrotome, let's talk about a regular microtome. A regular microtome, just the last part of the word, is shown here at the bottom right. And what it is an instrument used to make really thin slices of your sample. So usually what we do, for example, I used this when I was working with brain tissue in undergrad studies. So I took brain tissue and I embedded the brain tissue in some kind of wax. Then you basically use the microtome to make really thin slices so that you can look at them under the scope or do further staining. The microtome, when we cut these into thin slices, the thickness of each slice is in the units of micrometers. And this allows us to visualize them under the microscope, like a regular light microscope. An ultra microtome, as the one shown here, is very similar, except it's kind of what it sounds like. You're making extremely thin slices of your sample in preparation for electron microscopy. So instead of being as thick as what we produced over here, the thickness of these samples is going to be in the order of nanometers, so 1,000 times thinner than what we produced earlier. So staining and microscopy can be used for diagnostic applications. Our book gives us an example of diagnosing syphilis with microscopy. So here they have the bacterium Treponema pallidum, which causes syphilis, viewed under a dark field microscope. Here they're using bright field microscopy with a metal stain. On the right, it looks like they have looks like the surface. So it's a scanning electron microscope image of Treponema. And although EMs, electron microscopes, are not really used for diagnostics, it can help you further identify or characterize your bacterium. And remember, we can also use fluorescence microscopy. Use those labeled antibodies that are very specific to identify whether or not a specific bacterium like Treponema is present. In terms of immunofluorescence, in clinical settings, indirect immunofluorescence is often used to identify if that bacterium, Treponema, is present or not. So remember, for indirect immunofluorescence, what that means is the primary antibody does not have a label, right? See, there's no fluorochrome attached to that primary antibody. So if you just have that step, you wouldn't see anything under the fluorescent microscope. But the secondary antibody has the fluorochrome attached. So if the secondary antibody is bound to the primary antibody, then you'll get a signal. And remember that the signal is amplified when you have indirect immunofluorescence. And the reason for this is because multiple secondary antibodies can bind to each primary antibody, and that will amplify the fluorescent signal. That takes us to the resolution of that clinical focus problem from earlier. They did the gram stain. Remember, we had spherical purple cells. And after further testing, the lab tech determines that the actual bacterium causing the infection is Staphylococcus aureus. This is a biosafety level 2 organism that we're going to be working with in lab after the second week of class. She gets some antibiotics, and she is told to take up the entire dose, the entire course of antibiotics, to prevent reinfection and resistant bacteria. These days, antibiotic resistance is a growing concern. For instance, MRSA, methicillin-resistant Staphylococcus aureus, we know resists many antibiotics. It turns out that microscopy can help us in testing whether or not new antibiotics are effective. So our book gives us an example of a new antibiotic from marine organisms, bromophene, or MC21A. It just so happens that there's a green dye, SY2OX, Sidux, that stains dead cells. So if you're testing this antibiotic and you see that after exposure to the antibiotic, you see a lot of dead green cells, then you know that the antibiotic is probably effective. But we have to be careful in providing antibiotics widely. So these days, clinicians are usually much more hesitant in giving out or prescribing antibiotics than they were in the past. And this is because we have more and more superbugs these days, which are microbes that are resistant to almost all of the current treatments we currently have. So very thorough testing is usually completed before we prescribe antibiotics. You want to make sure you're not giving an antibiotic for a patient that's not going to be effective. But there's a lot of debate behind this. Patients can often get upset. Many patients don't know the differences between bacteria, viruses, and things like fungal infections. So they'll ask for an antibiotic and they won't understand that the antibiotic may be ineffective, right? And that's understandable from the patient view. I can understand why you go to the doctor. You want the doctor to do something to help you feel better, right? To get better. But clinicians and researchers and scientists in general really have to consider long-term public health risks, which is going to be something we're going to talk about again and more deeply in the middle of the semester. Let's recap 2.4 with the objectives. So simple versus differential stains. simple stains, they used a single dye, for instance, methylene blue, right? And the purpose was to highlight cell features like the shape, the size, or the arrangement of the cells. But remember, all cells are going to appear the same color. Differential stains use 2 or more dyes, and we're looking for differences between cells to characterize or identify the cell, or specific structures on or inside the cell. So differential stains included things like the gram stain, acid-fast, endospore, capsule, flagella. Unique features of commonly used stains. So examples like crystal violet, that was a basic stain, right? That's our primary stain for gram staining. Safranin. Safranin is a counter stain in both the gram stain and endospore stain. We had malachite green, right? Malachite green is good for staining endospores, but it requires that steam, that heat to penetrate the endospores. We had carbofuchsin, right? That penetrates the waxy cell walls of acid-fast cells. We had things like nigrosin for that background, the negative stain that stains the background but not the cells. And there are some more. Go back to the slides to look for those. Same thing for the last one, the procedures. So go through each of the staining procedures and make sure you can list the order of steps, what stains are involved, what color the cells appear after each step. So let's pretend we're going to do the acid-fast stain. So first step, right, primary stain was carbofuchsin, and both acid-fast positive and acid-fast negative cells will appear pink. Decolorizer was acid alcohol, and acid-fast positive cells will retain carbofuchsin. Acid-fast negative cells, the acid alcohol will wash out, and those cells will appear clear. The counter stain for the acid-fast stain was methylene blue. Methylene blue will not stain acid-fast positive cells. Remember, those are resistant to most stains because of that waxy layer, but methylene blue will stain the acid-fast negative cells. So in the end, for acid-fast staining, positive cells will appear pink, acid-fast negative cells will be blue. So go ahead and try the rest of these. Make sure to go through the steps. Look at that chart again. And that takes us to the end of Chapter 2. I'll see you next time in Chapter 3.